In this high-throughput screen for fungal endoglucanase activity in E. coli, the common microbial dye Congo red is used to visualize enzymatic degradation of carboxymethylcellulose by cells growing on solid medium. First, cells are transformed with an endoglucanase library and plated selectively for single colonies. Then, individual clones are picked and grown overnight in 96-well plates. Next, an enzymatic screen is performed by replica plating on CMC plates containing IPTG to induce endoglucanase expression. Finally, the active clones are picked from the master plate stored at -80 degrees Celsius. This assay produces unambiguous results as zones of degradation at the colony site, where halo size correlates with enzymatic activity and allows qualitative assessment of relative protein fitness.
The main advantage of this technique over existing methods, like the overlay method, is that it allows high-throughput analysis with a very low false positive rate.
Add 25 grams of LB growth medium, 15 grams of agar, and 1.5 grams of carboxymethylcellulose substrate to 1 liter of distilled water and autoclave to sterilize. After the medium is cooled, add appropriate antibiotics and IPTG to a final concentration of 100 micromolar. Now pour 200 milliliters of medium each into 5 large square Petri dishes or bioassay trays.